Assalamu alaikum and welcome everyone to the second part of molecular biology ASCP preparation series, Molecular Techniques. I'm Dr. Lai Abdelati and I'm really excited to share my knowledge and experience with all of you through this series. So let's dive in together. So today we'll continue in the fourth section of molecular techniques, nucleic acid amplification. Our focus for this season will be on PCR variations, a cornerstone of molecular biology diagnostic and high-yield topics for molecular biology ASCP exam. As we know, PCR or polymerase chain reaction is one of the most essential tools in molecular biology. It is what we use to amplify DNA. Okay? Over time, scientists have developed different variations of PCR to make it even more powerful. For example, real-time PCR allow us to actually quantify the DNA as it being amplified. Nested and semi-nested PCR improve specificity, helping us get more accurate results. Then we have multiplex PCR, which can detect multiple targets in one reaction. For absolute quantification, we turn to digital PCR. If we are working with the RNA, we use the reverse transcriptase PCR. And for detecting specific mutation, like SNPs, there is allele-specific PCR. Finally, arrays-based PCR let us screen many samples at once, great for high throughput work. So, each variation has a unique advantage depending on what kind of analysis we are doing.
Break it down small like DNA. Learning the code where secrets stay. Chromosomes dance under the light. Molecular dreams will take their flight. Molecular magic, it's in your hands. Unlock the science and take a stand. Study hard, let your passions ignite. ASCP success is within your sight.
Alright, now let's move to the question part and let us see the first question we have today. Alright, the first question we have today, which of the following steps is not a part of standard PCR cycle? As we know, PCR consists of three main steps. Denaturation, it means separating DNA strands, then followed by annealing step, it's binding primers, and extension, it is for DNA synthesis. Okay, so translation is the process in protein synthesis, not PCR. All right, let's move on. All right, the second question we have today, what is the typical annealing temperature in PCR? Alright, the annealing step where primers bind to their complementary DNA sequences usually occur between 50 to 65 degrees. Alright, let's move on. Okay, next question. Excessive PCR cycles often lead to. The correct answer is primer dimer accumulation. As the number of cycle increases, available DNTPs and template become depleted. Alright? These create conditions that favor interaction between primers, leading to the accumulation of primer dimers. Alright? Let's move on. Okay, next question. Which reagent is added to PCR master mixes to prevent carryover contamination from previous amplicons? The correct answer is DUTB. This nucleotide allows uracil to be incorporated into amplicons. A later reaction enzyme called uracil and glycosylase recognize and degrades this uracil containing amplicons. This process prevents the unwanted reamplification of carryover DNA, ensuring cleaner and more specific results. All right, let's move on. Here is the real-time PCR part. The first question, which component in real-time PCR allows for quantification of the target DNA during amplification? The correct answer is fluorescent probes. Fluorescent probes such as Tagman or Cybergreen emit light proportional to the amount of amplified DNA, enabling real-time monitoring and quantification. Other option, restriction enzymes, cut DNA, ethidium bromide, stain DNA post-amplification, and agarose gel is used for electrophoresis, right? So none of which allow real-time quantification. Let's move on. Okay, the next question. How does real-time PCR differ from traditional PCR? The correct answer, it detects amplified DNA in real time using fluorescence. Let's move on. Next question. Which of the following is the common method for detecting DNA in real-time PCR? The correct answer is cybergreen fluorescence. Cybergreen fluorescence binds to the double-strand DNA, emitting fluorescence proportional to the amount of amplified DNA in qPCR. All right, let's move on. Okay, the question here, what does a low CT value indicate in real-time PCR? The correct answer is high template quantity. Actually, a low CT value means the fluorescent signal crosses the threshold early, right? Which indicate a high abundance of starting DNA in the samples. So, if the template quantity were low in option A, the CT value would be higher as it would take longer for the signal to reach the threshold, while efficiency in option C and contamination option D can influence CT value. They are not the primary indicator of high template quantity. Okay, let's move on. All right, the next question here. Which feature of real-time PCR distinguishes from the conventional PCR for quantification? The correct answer is continuous fluorescent monitoring. Actually, real-time PCR allows DNA quantification as the reaction progresses using fluorescent signals to track amplification at each cycle. In contrast, conventional PCR detects DNA only at the final stage, making it less precise for quantification. While DNTB's option A and amplicon size option D are essential for PCR, they do not distinguish real-time PCR from conventional methods. All right, let's move on. Okay, the next question here. What is the purpose of a melt curve analysis in real-time PCR? The correct answer is to verify amplicon specificity, all right? Actually, multi-curve analysis helps confirm the specificity of the amplified DNA by examining its denaturation temperature. So each DNA fragment has a unique melting point, and distinct peaks indicate correctly amplified targets. While CT values are used for quantification in option A, multi-curves primarily serve to check the accuracy of the amplification. All right, let's move on. Okay, the next question. A multi-curve with the multiple peaks suggests the correct answer is non-specific amplification or primer dimers. All right, let's move on. Okay, the next question here. What is the primary purpose of including a passive reference dye such as the ROX in real-time PCR? The correct answer is to normalize fluorescent fluctuation between wells. Actually, ROX dye provides a stable baseline fluorescent, helping to correct variation in reaction volume and instrument optics between wells. This ensures consistent and reliable measurements, okay? It does not stabilize enzymes in option A or interact with the primers option C and D. All right, let's move on. All right, the next question here. Why is the standard curve required in absolute quantification using qPCR? The correct answer is to correlate CT values with known DNA copy numbers. Actually, a standard curve is created using serially diluted DNA samples with known concentration. This curve allows researchers to link CT values to exact DNA copy numbers, making it possible to quantify unknown sample with accuracy. All right? While fluorescent option A and primer efficiency option D are important for PCR performance, they are not the primary purpose of the standard curve. Let's move on. Here is the nested and semi-nested PCR part. So let's see the first question here. What is the main advantage of nested PCR? The correct answer, it increases specificity by using two sets of primers. Actually, in PCR, enhance specificity by using two sets of primers in consecutive amplification rounds. The first round amplifies a border target region, while the second round, using internal set of primers, focus on more specific segments. This approach significantly reduces non-specific amplification and improves sensitivity, making it ideal for detecting low abundance target in complex samples. Let's move on. What distinguishes semi-nested from nested PCR? The correct answer is use three primers total. Okay. Semi-nested PCR uses one original primer and one new primer in the second rounds, in total of three primers, while nested uses four primers. Okay. Actually, both require two rounds in option B, but primer count distinguishes them. Okay, let's move on. Please, if you found this content helpful, don't forget to like, share, and subscribe. Your support means the world to me and help me create more valuable content for you. Thank you for being part of this journey. Let's continue. Okay, next question. Why is the second round of PCR performed in nested PCR? The correct answer is to enhance specificity. Actually, nested PCR improves specificity by using 2 rounds of amplification. In second round, primers bind within the first PCR product, effectively reducing non-specific binding and unwanted amplification. All right, let's move on. Okay, the next question here. Why is nested PCR commonly used in pathogen detection? The correct answer, it eliminates false positive by increasing specificity. As I said, nested PCR enhances specificity, making it useful for detecting low abundance pathogen while minimizing false positives. All right, let's move on. Okay, the question here, what differentiates semi-nested PCR from fully nested PCR? The correct answer is only one primer is replaced in the second round. As I said, in semi-nested PCR, one of the original primers is retained while a second inner primer is introduced in the second amplification round to improve specificity. All right, let's move on. Okay, next question here. Why are two separate primer sets required in nested PCR? The correct answer, to reduce the specific products from the first PCR. Actually, the second primer set targets a sequence within the initial amplicon. This ensures that only the desired specific product are amplified in the second round, while non-specific fragments from the first PCR are minimized. By focusing on a smaller, more precise region, the reaction improves specificity and accuracy. All right, let's move on. Okay, let's see the next question here. Why is the product of the first PCR not visualized on gel before the second round in nested PCR? The correct answer, to reduce contamination risk. All right. In PCR workflow, opening tubes to check the first product can lead to aerosol contamination, which may compromise the accuracy of the result. Nested PCR avoid this step entirely, helping to maintain a clean and controlled environment through the process. All right, let's move on. Now we move to multiplex PCR questions. The first question here, what is the main advantage of multiplex PCR? The correct answer, it amplifies multiple DNA targets in a single reaction. As we know, multiplex PCR allows for simultaneously detection of multiple genes, saving time and reagents. All right, let's move on. The next question, what is the major challenge in multiplex PCR? The correct answer is primer dimer formation. All right, when multiple primers are used in a reaction, there is a high chance that will bind to each other instead of to the target DNA. This unwanted primer interaction, known as primer dimer, can compete for reagent and reduce the efficiency of the PCR, sometimes even causing failed reaction. Okay, where factors like sensitivity in option A and cost option D are less important in this case, and the time required remains similar to standard PCR. All right, let's move on. Okay, the next question here. Which step is critical for optimizing multiplex PCR? The correct answer is using identical prime melting temperature. Okay, when primers have a similar melting temperature, they anneal efficiently at the same temperature during PCR. This consistency ensures that the both forward and reverse primers bind properly, leading to accurate amplification. Okay, if the melting temperature value differs significantly, one primer may anneal while other remain ineffective, reducing reaction efficiency, right? While other option, like using excess enzyme in option B or magnesium chloride in option D, could instead increase the chance of non-specific binding rather than improving primer annealing. Okay, next question here. Which procedure adjustment is the most critical when designing primer for multiplex PCR? The correct answer is avoiding complementary regions between primers. All right, let's move on. Okay, the next question here. What is the common indicator of failed multiplex PCR on a gel? Okay, the correct answer is the smear-like staining across the lane. Actually, a smear on the gel suggests non-specific amplification or primer dimer formation. This often happens due to poor primer design or competition between multiple reaction components. So, if the gel shows a single band, it means only one target was successfully amplified. No bands indicate a complete failure of the reaction. Okay, let's move on. Now we move to the PCR arrays. So the first question we have here, which application is best suited for PCR arrays? The correct answer is high throughput gene expression profiling. Okay, let's move on. Okay, the next question here. PCR arrays are commonly used to screen for. The correct answer is hundreds of cancer-related mutations. Actually, arrays are powerful tools for detecting many genetic mutations at once, making them ideal for high-throughput screening of cancer-related mutations. This allows researchers to analyze a large number of mutations efficiently, which is essential for studying cancer genetics. All right? So, why not SNPs? SNPs, as we know, is a single nucleotide polymorphism, are better detecting using allele-specific PCR, a more precise method for identifying individual genetic variation. Okay, so why not whole genome methods? In option C, whole genome techniques focus on the sequencing the entire genome, which is the broader approach than necessary for targeting mutation screening like cancer panel. Okay, so the correct answer here is hundreds of cancer-related mutation. Let's move on. Okay, next question here. What is the final step in array-based PCR before data analysis? The correct answer here is hybridization of amplicons to probe-coated chips. As we know, PCR products bind to the complementary probes on the array for detection. Okay? Other options, staining option B and gel option C are used in conventional PCR, not arrays. Okay? Let's move on. That's all for today. I hope you enjoyed this lecture and look forward to hearing from you what you think of it. Please don't forget to like, comment if you have any questions, share and subscribe to our channel to receive all the latest lecture. And don't forget to click here to watch the previous video. That's all. Salam.